Hi, my name is Mimansa Dogra. I go to Interlake High School, and today I will be talking to you about Rett syndrome and finding a cure for Rett syndrome through gene therapy. So, what is Rett syndrome? So, Rett syndrome is a neurological genetic disease that is caused due to a mutation on the gene MECP2, which is located on the X chromosome. It is fatal in cases of boys, since boys only have one copy of the X chromosome, but most girls survive. Symptoms of Rett syndrome include mental deficiencies, stunted growth, poor motor skills, and abnormal head growth, as well as blindness and in some cases loss of use of limbs. So anyways, here's a question that most people have when they first hear Rett syndrome. Why do girls survive? How are they protected from lethality when they acquire Rett syndrome? So girls survive due to a process called X chromosome deactivation. It is a process in which one X chromosome is randomly deactivated. So as you can see here, I have made a couple of cells and I have X chromosomes in each cell. So let's assume that the blue X chromosome is a maternal X chromosome and the red X chromosome is a paternal X chromosome. So let's assume that the paternal X chromosome is the X chromosome with the mutated MECP2 gene, while the blue X chromosome is the X chromosome with the normal MECP2 gene. So I'm going to simulate X chromosome inactivation here. So you can see that some X chromosomes are randomly deactivated. So, as you can tell, there are some X chromosomes which are expressing the mutant MECP2 gene, and there are other X chromosomes which are expressing the normal MECP2 gene. And in cases of girls, the expression of some normal MECP2 genes is apparently enough for survival. However, they do suffer from lifelong symptoms, which I have described before. So the question is that how can we prevent these symptoms from occurring? So obviously we cannot rewrite their DNA and take out genes and put back in genes or do anything of that sort, but how can we prevent the symptoms of Rett syndrome? How can we make their standard of living better? So here's another picture of X chromosome deactivation. This is probably one of the better known examples of X chromosome deactivation. This is called a calico cat. Now, as you probably know, calico cats are female because of the X chromosome inactivation process. Calico cats are special in the sense that they have very particular color markings. You can see some of the patches of fur are black, some are orange, some are white. These are all caused by X chromosome deactivation. So, for example, in cases of black fur, the X chromosome with the gene which carry the allele for orange fur has been deactivated, while it's vice versa for the orange fur. So, and then that also explains why calico cats are primarily female, because of this process of X chromosome deactivation. So we've talked enough about that. So let's talk about what causes it, and that is primarily caused by cyst. So cyst is a gene that is responsible for random X chromosome deactivation. So without cyst, the X chromosome cannot be deactivated. However, many other genes are needed to aid cyst expression, or in other words, help cyst deactivate the targeted X chromosome. Now here's something important. Cyst is needed in order to deactivate the X chromosome. However, let's say like gene X and gene Y, they by themselves cannot deactivate the X chromosome, but they are used to help cyst. Cyst by itself does a very poor job of deactivating the X chromosome and needs the help of gene X and gene Y. So cyst is a main gene here. So we're going to look at one of these genes which aids cyst in deactivating the X chromosome, gene 1. We're going to call it that. And so gene 1 is one of those genes which aids cyst, and one of the genes that are partly responsible for X chromosome deactivation. So this is going to be important because we want to see like whether we can knock down gene 1. So shRNA, which are small hairpin strands of RNA, are used to knock down genes or prevent them from expressing themselves. So the genes are there, they are present in your DNA. However, they are not able to express themselves to create mRNA or create proteins, which eventually lead to symptoms of different syndromes such as Rett syndrome, or even your eye color, your hair color. So basically, if we were to knock down cyst, then it would not be able to express itself. So the targeted X chromosome would be expressed because cyst has been knocked down. However, an easier way to do that would be to knock down the genes that assist cyst, like gene 1. So, if gene 1 assists cyst, then if you would knock it down, then would the cyst expression decrease? Well, this is the question we are trying to answer in this experiment. So my hypothesis is that if gene 1 was knocked down, then the expression levels of cyst would also decrease, because gene 1 is needed to help cyst express itself. If you knocked it down, then the levels of cyst would decrease, and therefore the expression levels of X chromosome, the targeted X chromosome, would thereby increase. So, in order to successfully complete my experiment, I had to use a process called qPCR. And qPCR is called real-time polymerase chain reaction. It is a process used to measure gene expression and is able to replicate a gene of interest outside the human body. So basically, we attach primers to a gene of interest and we replicate it under certain conditions. And under those certain conditions, we are able to see whether the gene of interest is able to express itself fully or not. Now, on the left is a qPCR machine. Yes, its name is George. This is a standard qPCR machine. This is the one I used for my experiment. And then you can see over here, here is the plate well I used for my experiment. So I will be talking about procedure next. So the qPCR, basically what you do is that you heat up the DNA until you denature it, and then you use proteins to kind of do a mock DNA replication trial, except you're only targeting one gene. So after you replicate it, after 40 cycles of replication, of course DNA increases exponentially since you're replicating each, because it's pretty much given information that DNA replication is an exponential growth process. You are able to see the expression of the gene of interest. So I'll be talking about the procedure next, and the procedure is kind of complicated, so I have simplified it a little bit. So the procedure is that we set up two main qPCR trials. One would see DNA that had gene 1 knocked down. That's a manipulated trial. And one would gene 1 as it was, which is a control trial. We use a fluorescent dye to bind itself to the DNA, place primers around the gene of interest, cyst. We then observe whether the expression levels of cyst changed at all. So we basically set up a standard qPCR trial, 40 cycles of DNA amplification, and we had knocked down cyst, and then we also attached primers to the gene of interest, and all of these were in the qPCR mixture which we used. It was basically a standard mixture, and we also used a GAPDH gene, in order to have an internal control, but we'll talk about that later. So that's our procedure. It's pretty simple. I've simplified it quite a bit. And what you get is that, let's see if you can zoom in here a bit. Okay, so what you get is that for the control trial with cyst and gene 1 being as it is, the expression was 100%. So with gene 1, cyst was able to express itself fully. However, in the bottom two cases in which we did our manipulated trial, gene 1's expression rate was only 85%. So with the absence of gene 1, or rather with gene 1 knocked down, cyst expression rate, was only 85%. And how do we know this? Well, we looked at a control gene, GAPDH. So GADPH, is an internal control. And what's really interesting about this kind of gene is that it behaves similarly in every type of situation. So no matter what tissue or what membrane or what cell you put this gene in, it basically behaves the same. So it's a nice internal control for a qPCR experiment in order to normalize the results and actually see how the expression levels have changed. So I already mentioned that cyst expression when gene 1 was knocked down went down to about 85%. So as shown from the table, when gene 1 was knocked down, cyst expression drops to 85%. Therefore, my hypothesis was proven correct. Gene 1 is important to regulate cyst expression and thereby is important for X chromosome deactivation. So therefore, if you knock down gene 1 and cyst expression goes down, then the X chromosome is reactivated. And now we're coming to the question, well, how is this important? Why should I care? Now think about this. We have 2 X chromosomes, one with a mutated gene, MECP2, and one with a wild type, which is a normal MECP2. So if we are able to reactivate the X chromosome with a normal MECP2, we are probably able to alleviate some of the Rett syndrome symptoms. So that would be a big breakthrough, because remember I told you we cannot rewrite the DNA. We can only work on the symptom level. But that would be a big improvement. We're working on the molecular level. So in order to do this kind of gene therapy, much more research has to be done in this area. If we are completely able to suppress the cis gene and allow the chromosome to express itself, we have not taken into account the other X chromosome. So I'm kind of getting ahead of myself here, but I'm just going up to the hypothetical scenario in which you are able to completely reactivate the other X chromosome with a healthy gene MECP2. However, the other X chromosome is not deactivated. So now in a single cell, you have 2 active X chromosomes. So that could lead to potentially dangerous and unknown situations, frankly. So a lot more research has to be done in order to test out this type of gene therapy, turning on and turning off X chromosomes. But even though so much more research has to be done in this area, learning about gene 1 and the ability it has to regulate cyst expression is an important step in learning how to alleviate Rett syndrome. So while these are just small steps, these are important steps to being able to solve the larger picture. So therefore, since we have figured out that gene 1 is important to Rett syndrome, you could say that we are that much closer to being able to completely cure the symptoms. So thank you for listening to me. My name is Mimansa Dogra and I'm from Interlake High School.